Assalamualaikum and hi everyone. I am representative from Group 5 and today I will present about gamma irradiation induced grafting of 2-hydroxyethyl methachloride, or we call it HEMA, onto EPTFE for implant applications. So our journal was published on 20 March 2020. So firstly, I will give a brief introduction for our study. In this study, EPTFE is used in this study, and EPTFE has a very high hydrophobicity, and also various techniques has been used to alter the chemical and physical properties of this polymer surface. HEMA was chosen as the grafted monomer. So EPTFE was evaluated using various characterization techniques such as ATR, FTIR, spectroscopy, XPS, EDX, and many more. And then, the effect of grafted EPTFE is tested by using simulated body fluid, SPF, that used to miming the solution in human body. Next, I will continue to the material used in this study. So, first thing first, we have to have an EPTFE membrane, and the methanol from Merck and HEMA, ammonium iron 2 sulfate hexahydrate, the ionized water, and for the protein absorption study, we must have ovine serum albumin, KH2PO4, Na2HPO4. And then for stimulated body fluid, things needed are NaCl, HCl, NaHCO3, K2HPO4, TH2O, and many more. Next, we will move into procedure. So first thing first, we have to prepare the monomer solution, right? So the ingredient needed is various concentration of HEMA, which is 2%, 10%, 20%, and 30%. And also, we need 20 mg of more salt. So the purpose of it to form the monomer solution. And secondly, we have to prepare the EPTFE sample. So we must make sure that the dimension is correct, and also the dumbbell shape. And also the action is we need to cut and immerse it in the monomer solution. And after that, we need to degas it. We need to cover the test tube with the paraffin and then degas it with nitrogen for 50 minutes to push oxygen. And fourth, we need to irradiate it with the condition as written in our mind map, which is temperature is 25 degrees Celsius and radiation type used is gamma ray, and the doses needed is 2 kg, 5 kg, 10 kg, and also 20 kg, and the dose rate is 1.767 kg per hour. And then we have the post-irradiation washing. We need to do the initial wash, subsequent washes, and overnight soak. And after that, we dry it. We dry it in the desiccator jar until a stable weight was achieved. And after that, we replication, parameter recording, which is each parameter was recorded in triplicate. And then sample naming convention in the format. And for example, sample irradiated at 2 kg and grafted in 10% concentration of HEMA is labeled HEMA2 in the bracket 10. So meaning D mean dose of irradiation and M mean concentration of monomer. Alright, next we will move to the procedure for the stimulated body fluid study and also protein absorption study. So for the stimulated body fluid study, firstly, we need to immerse the untreated and grafted sample in the 1.5 times SPF solution at 36.5 degree for 2 weeks. And after that, the solution was changed at interval of every 3 days and no change in the pH was observed. And after the 2 weeks, the sample were removed and dried in the desiccator jar until the constant weight achieved. And after that, the decomposition of CAP minerals onto the EPTFE surface was measured by the increase in weight. Alright, so for the protein absorption study, the protein absorption study solution was prepared by dissolving NaCl, KH2PO4, and Na2HPO4 in the 800 ml of deionized water at 36.5 degree in the plastic beaker. And after that, after we dissolve the reagent, 20 ml of the deionized 200 ml of the deionizing water was added. And then the pH of the solution is measured at all time during the preparation, and the pH of the dissolved reagent was adjusted to 7.5 using NaOH and also HCl. And after that, the untreated and grafted sample were immersed in 15 ml PBS solution containing 1 mg of PSA for 1 hour at 37 degrees, and the sample were then washed with deionized water and dried in the desiccator jar until a stable weight was achieved. So this is how the procedure for the protein adsorption study and also for the stimulated body fluid study. Alright, so we will move to the characteristics. So what characteristic of HEMA that we can focus on is, first thing first, we can look at the grafting yield determination. So the method used is we measure the weight changes before and after, and then the equipment needed is electronic balance, and we can see actually the average and the standard deviation in the triplicate. And the secondly, we can watch water uptake, and the method used is we immerse EPTFE sample in the ionized water for 24 hours, and then we brought excess water with filter paper, and then after that we weigh it, and then we can see the average and standard deviation in triplicate. Third is contact angle measurement, which is the equipment needed is automated angle gyrometer machine, Rama Hart, and then we can see the average and the standard deviation. After that, also we can see the chemical composition analysis using ATR FTIR spectroscopy. And then we can see the peak refinement based on the previous published work, and then for the morphology analysis we can use FESEM or EDX analysis, and then for the mechanical properties testing we can use tensile test. So the equipment needed is universal tensile machine, and then we can see the average plus minus standard deviation for at least 5 reading. Next, for statistical analysis, it is conducted between two independent samples for analysis for mechanical properties and water contact angle. So a t-test was performed for this specific comparison, and the statistic mean was based on the p-values of less than 0.5. So we will go to the effect of grafting parameters to HEMA. So firstly, grafting yield are affected by radiation dose and also concentration of monomer. So for radiation dose, the study found that the increase in the radiation dose, the radical that are involved in the grafting reaction also increase. And then for the concentration of monomer, the increase in the concentration of HEMA most likely result in the higher diffusion of the HEMA concentration in the grafting region. And the effect of grafting to HEMA is, firstly, we can increase in dose dependent manner, which is the grafting reaction occur initially at the surface of the sample and progresses inward as the grafting zone is swollen by the monomer solution. And second, our monomer become very hard and brittle. For further increase in HEMA concentration resulted in the further homopolymeter formation, thus hindering the diffusion of HEMA onto EPTFE. And then it's also increased the ability to sustain water uptake. The high water uptake and decrease in the water contact angle is attributed to the hydrophilic component of HEMA, which become hydrogel after irradiation. That's all from us. We hope that you guys can get new knowledge about gamma irradiation induced grafting of 2-hydroxyethyl metachloride HEMA untuk EPTFE for implant applications. And thank you for listening.